I'm Arlene Montalvo. I'm the president of the Riverside San Bernardino chapter, and I welcome you here today to listen to a couple of really cool presentations that will be introduced by Orchid in a couple of minutes. I just wanted to remind you that we're hosting this from the Riverside San Bernardino chapter of CNPS. We are one of 26 chapters throughout the state. Membership now in just over 12,000, which is, so we're really growing. CNPS is doing really well. We do a lot of conservation activities and try to preserve and promote the wonderful flora that we have here in California. One of the things that we're trying to do this spring, since it's going to be, it's turning out to be a pretty darn wet year, as you know, and there's a lot of wildflowers out there. We have organized a number of field trips, so be sure to go to our website, Riverside-San-Bernardino-CNPS.org, and you'll see a little calendar on the field trip page, just a listing of the field trips with their dates. And as time goes by, we will be adding flyers with details for those field trips, so don't miss those. We also have some real dynamite presentations coming up on the third Saturdays of the month at 10 a.m. So be sure to look at the calendar of events that we have posted. You have to actually do a click on the homepage, I think, to download the or see the picture of the calendar of events. So there's a lot going on. If you're not a member of CNPS, please do consider joining. You can go to cnps.org and join online. We may have some promotional memberships coming up for some events. So we will hopefully have you join in with us. So I am going to go ahead and introduce our program coordinator, Orchid Black. She was stuck in Crestline a bit recently.
In Arrowhead.
In Arrowhead, yeah. In the snow, but she's finally out and was able to get down the mountain. So I've been to Orchid Black. Thank you.
Welcome, everyone. Before I introduce the speakers, I would like to introduce Katie Barrows, who is our remote host. And she'll tell us how to deal with the questions from the online users.
Good morning, everyone. Thanks, Orchid. Can everybody hear the audio okay? There was a comment in the chat about the audio. Hopefully everyone's able to hear. I just want to let you know we're going to ask that you hold your questions until the end of the presentations. I guess maybe we could do questions after the first presentation, Orchid, and then follow the second one. Does that make sense? And if you would please put your questions in the Q&A, and I'll be monitoring those. And once the speaker is finished, I'll ask the questions. So there'll be plenty of time to ask questions, but please put them in the Q&A. I'll monitor the chat as well, but we prefer that you use the Q&A. Thanks very much. Enjoy the presentation.
Thank you, Katie. I'm just going to do a little bit of cleanup here, and then a little bit of tech things. I want to say that hybrid meetings are still new to us, and though we would like to think that we can provide a smooth experience in the room and to the Zoom, that's not always the case. So feel free to chat, or feel free, if you're in the room, to come and get me, but we apologize in advance for that. It's probably not going to be. And then I would like to introduce our wonderful speakers for today. We have two fifth-year PhD students in the UC Riverside Department of Evolution, Ecology, and Organismal Biology. Say that 3 times fast. And they are both studying plant-pollinator interactions. And first we will have Annika Roseperson, and she's studying plant-pollinator interactions in alpine ecosystems and locally. And then we will have Elijah Hall, and he's studying plant-pollinator interactions along climatic gradients in dryland environments. So welcome, Annika.
Hi, thank you for that introduction. Can everyone hear me okay on Zoom?
Yes, we can hear you.
Okay, great, thank you. Just want to make sure my audio is coming in loud and clear. So yeah, thank you for that introduction. It's great to be here. Thank you so much for having me. Yeah, my name is Annika. My pronouns are she, her, hers, and I'm in the Cool Rafferty lab at UC Riverside. And I'm excited to share with you about one of my dissertation chapters today, where we're looking at how drought is impacting plant traits and also plant-pollinator interaction. Here we go. Pollination is a mutualism that's critical to ecosystem functions. So up to 88% of flowering plant species actually rely on animal pollination to reproduce. And this animal pollination not only helps this almost 90% of plant species create seeds, but it also helps them outcross. So this allows them to increase genetic diversity. Increasing genetic diversity is going to become especially important as climate change starts to alter the environment that these plants have to live in. And on the other side of this mutualism, over 1 million insect species rely on the floral resources that plants provide to them. So importantly, insects are being threatened by global change drivers like climate change and land use change. So understanding how these interactions occur and how they're impacted by climate change is becoming more and more important. So for this really important mutualism to occur, there are a few conditions that have to be met. First, the plants or their flowers and pollinators have to occur at the same time in space, so across the landscape, and at the same time phenologically, right? So in the same time frame. And then to get pollinators to visit flowers, plants create signals. So these signals can be visual, like by producing flowers that are large, showy, by producing a lot of flowers, like the ones pictured here on the left-hand side of the screen. This is from the Super Bloom on the Borrego in 2019. They can also produce like showy, bright colored flowers that attract pollinators in, right? But they can also use non-visual cues, like olfactory cues or scent compounds that help attract pollinators at different times of day. So in tandem with these signals, plants provide pollinators with floral rewards. I'm going to talk about two main floral rewards today, which are nectar. So nectar is a sugar slurry. It's a combination of like sucrose, glucose, a few other types of sugars, and also sometimes with amino acids and water. And nectar is used for basic energy use throughout the day for bees and other pollinators. Pollen is another critical floral reward. It's mostly like proteins and fats. So it's kind of like the protein bar that the plant is offering to the pollinator. But it also serves the important role of like the reproductive currency of the flower, right? That's the male gamete. And so when the pollinator collects that pollen, it's eating some of that pollen, it's providing some of it to its offspring in some cases. And it's also sometimes transferring that pollen to another flower, which allows those plants to reproduce. And together, these signals and floral rewards influence the behavior of the insects that are pollinating plants and other organisms that pollinate plants. This behavior can include things like choosing which flower to land on, choosing how long to stay on a single flower, how often to move between different flowers, and they can even influence the memory of insects as well as other pollinators. So for example, some bees can actually remember which flowers are going to be the most nectar or pollen rich for them, return to those same flowers again, or the same species again. And in turn, this pollinator behavior provides the plant with pollination services, which just means that the pollinator helps those plants reproduce and helps those plants have more offspring and become more prevalent or maintain the population size. However, as you are all likely very aware, droughts are predicted to increase in severity and frequency due to climate change across different parts of the globe. California has experienced its fair share of droughts over the past few years. We've certainly, had much less of a drought year this year, which has been terrific. But in certain parts of the globe, like the Mediterranean, for instance, droughts are predicted to become much more severe and much more frequent. Unfortunately, this drought impacts plant-pollinator interaction. So this can be happen through a couple of different pathways, right? So first, drought can impact the signals that plants provide to pollinators, so like the size, number, shape, color of their flowers, and it also impacts the floral rewards of those flowers are attracted for insects. And this has an indirect effect on the behavior of those insects and other pollinators, which ultimately impacts the pollination services that those insects are providing. So with all of this in mind, I wanted to ask a couple of main questions. So first, how is drought stress impacting floral traits? So those things like signals that bring pollinators in and the resources that flowers provide pollinators. And our second question, we asked how those changes in traits impact pollinator foraging behavior and their foraging preference. So to answer these questions, we're using a couple of focal species. We're working with baby blue-eye for Nemophila rosezii and desert bells or Phacelia concatenaria. They're both in the borage family. They're both native to California, and they've both been visited by our focal insect species, who I'll introduce in a few slides here. Nemophila grows throughout California. It's generally more, it tends to grow more like between trees and shrubs, whereas Phacelia is more focused in on Southern California, right, kind of right around Riverside area and a little south of us. And it tends to grow in sandier, drier soil areas. And one thing that I really want to talk about these two species is their turgor loss point or their drought tolerance, right? So this is the first of many figures I'm going to show you today. In this figure, as I will be throughout the rest of the figures, I'm showing the Nemophila on the left-hand side and Phacelia on the right-hand side. My y-axis is showing turgor loss point. So this turgor loss point is just a metric of how drought tolerant plants are. And as TLP or turgor loss point becomes more negative, that means the plant is more drought tolerant. We can see that Nemophila has a wilting point or turgor loss point of like -0.6, and Phacelia's is more around -1.2. So this tells us that Phacelia is quite a bit more drought tolerant than Nemophila is. Anybody who's grown these in their yards probably is already aware of that fact, but this is an important thing to keep in mind as we move forward with the presentation. To answer our first question, we grew a bunch of plants in a greenhouse in the years 2021 and 2022. Half of them were given what we call drought conditions, and then half were given adequate water, just ample water. And then we measured a number of traits. So in 2021, we measured the number of flowers and the size of those flowers. And then we also took measures of nectar volume and nectar sugar concentration. In 2022, we measured all of those same things, but we wanted to add a little bit more. So we looked at also water potential, which is a measure of how drought stressed plants are. And then we added this metric of nectar refill volume and nectar refill concentration. So whereas in 2021, what we were measuring was just the nectar volume and sugar concentration of a single flower once per day, what we added in 2022 by measuring nectar refill is how much nectar can be replaced in that flower within a single day after nectar has already been removed one time. So this nectar refill was measured in the afternoon, whereas the initial nectar volume was measured right in the morning from that flower. So let's start off by talking about how successful our treatments were, our drought treatments were. So again, I'm showing Nemophila on the left-hand side of this figure, and I have Phacelia on the right-hand side of the figure. And on my y-axis, I'm showing leaf water potential, which is again a metric of how drought stressed a plant is. And as this number gets more negative, so as this y-axis decreases, that means that the plant is more drought stressed. Okay, so in the morning, we found that adequate watered plants, which are shown in this light turquoise color, are less drought stressed than the droughted plants, which are shown in the dark orange color. We found the same pattern in the afternoon. So this is just telling us that our treatment was effective. So just kind of like a sanity check to make sure that our treatment's working. So now that we know that our treatment's effective, thank you. We can start talking about how that drought treatment actually affected the plants, right, and their traits. So first, let's talk about floral abundance. So it turned out that in both Phacelia and Ammophila, again, Ammophila on the left, Phacelia on the right, we had way more flowers per plant in adequate water. So plants with given adequate water are producing like 2 to 3 times as many flowers in both species. And then if we move on to floral size, we see a similar pattern where adequate water plants are producing flowers that are like 2 to 3 times larger in terms of both the floral landing area, which is kind of the surface area at the top of the flower, and also the height of the flower or the corolla length. So we're producing plants, or we're producing more flowers that are larger. Now let's move into some of those floral rewards that plants are providing for pollinators. So I'm going to be showing you data from both the morning and the afternoon. So I'm looking at both the initial nectar that the plant was producing in the day, and then also that refill nectar that the plant produced to refill its nectar stores in the afternoon. So we found that in the morning, adequate watered Ammophila plants produce significantly more higher volume of nectar than droughted Ammophila plants. However, in the morning, Phacelia plants produced the same amount of nectar for both the adequate and droughted plants. So regardless of treatment, they're producing a pretty substantial amount of nectar. Moving into the afternoon, though, the refill nectar for both Ammophila and Phacelia was much higher in adequate watered plants. And then we can also look at nectar sugar concentration. So we found that in both the morning and the afternoon, for both Ammophila and Phacelia, droughted plants were producing more concentrated nectar that just had more sugar. So this is likely just because there's less volume of nectar generally, and so the sugar is just more concentrated. This is a lot of information, but what I really want to point out is this discrepancy here, where in the morning, the Ammophila was producing a greater amount of volume in the adequate water plants, whereas in the morning, Phacelia was producing the same amount of nectar in both the adequate and droughted. And this pattern is going to be important when we move on to talk about how pollinators are responding. So to summarize, for Ammophila, we saw that adequate water plants produced more flowers that were larger, and these flowers produced more nectar that was less concentrated in both the morning and the afternoon. Phacelia plants, for Phacelia plants, the adequate watered individuals produced larger flowers and more of them. The nectar volume was not different between adequate water and droughted plants, however, in the morning, but in the afternoon, the adequate water plants produced more nectar that was less concentrated than the droughted plants. Okay, so that's kind of our answer to question 1 about floral traits, but let's move on to question 2, where we ask how drought stress is affecting pollinator behavior and pollinator preference by altering this floral trait. Again, we're working with Ammophila and Phacelia, but we're now introducing this wonderful pollinator, Osmia lithariaceae, or the blue orchard. The megachilid bee, and it's terrific. It's like a super adorable little guy. It lives in the western half of the United States. And it has this really interesting reproductive system where the females emerge in the spring. They collect a bunch of pollen and nectar and create these little pellets of pollen that you can see here. And then they lay an egg next to that pellet of nectar pollen, and then they seal off the chamber with a little dab of mud. And then they repeat this process like 30 to 40 times throughout the course of the summer. And then these eggs hatch. The little pupa eats the pollen and nectar provision and pupates into an adult over the winter and emerges in the spring in the process. And so these pollinators have been shown to be really important pollinators in the natural system of native plants, but they're also becoming a more and more important agricultural pollinator. And I bring this up because, as some of you may know, Apis mellifera, the European honeybee, is a non-native pollinator that's commonly used in agricultural settings. And it has some pretty negative implications for native pollinators and for native plants. And so shifting to using native pollinators in agricultural settings can kind of ameliorate some of these issues with using basic pollinators. So the more we know about osmia, the better. Right. So to find out how osmia responds to drought, we used these choice trial chambers and again grew up all of our plants And so to look at preference, we started off in the morning with a freshly born female osmia. So she's totally naive, hasn't experienced anything in her life yet. We put her in one of these chambers, and then we grabbed a droughted plant and an adequate water plant. We measured some traits from them, and we bagged all but 5 flowers so that her behavior was only being impacted by the traits on the flowers, not just the number of flowers. We popped those plants into the choice trial chambers, and then we observed and reported her behavior for 15 minutes. Then we came back in the afternoon, grabbed the plants out again, measured traits again, bagged flowers again, and put the plants back in the cage and performed our afternoon 15-minute observation of her behavior. So I only have one figure to show you from this part of the experiment. And again, we've got Collinsia on the left with the sweet little osmia foraging on that flower, and also Phacelia on the right. So in this figure, I'm going to be showing you the preference for adequate watered flowers. So we're on the y-axis looking at the percent of time that each female spent foraging on adequate watered plants of her total foraging time. So if the values are above this dotted 50% line, this means that she was showing a preference for adequate watered flowers. And if the values are below the dotted line, that means she's showing a preference for droughted flowers. And I'm going to be showing you both morning and afternoon observations at the same time. The morning is going to be in that dark blue, and the afternoon will be in this light yellow. Let's start with Nemophila. So for Nemophila, we found that osmia significantly prefer adequate watered plants in both the morning and the afternoon. They're choosing to forage on the adequate water flowers around 80 to 90% of the time, and their preference doesn't shift between the morning and the afternoon, right? At both time points, they're preferring those adequate water flowers like consistent. However, when we look at Phacelia, we find that the pattern's a little bit different. So in the morning, osmia are showing a slight preference at around like 62% for adequate watered flowers. Our p-value was a little bit above significance level, so it's a marginally significant trend. However, in the afternoon, we see a strong and much higher preference for adequate watered flowers. So why is there this big shift? Oops. Sorry, I don't know. That was strange. Okay. Well, I will preview for you there. Why was there this big shift in preference in the afternoon in Phacelia? So why did they start out in the morning not really caring which flower they foraged on as much, and in the afternoon they started really preferring this adequate water? Well, I'm going to bring up this figure again that I kind of highlighted earlier in the talk that shows the nectar volume between the morning and the afternoon for Phacelia. So you'll recall that I mentioned how in the morning, adequate and droughted Phacelia plants produced around the same amount of nectar, whereas in the afternoon, the droughted plants produced significantly less nectar, right? So what we think is happening is that the pollinators are picking up on this trend, and they're really responding to the volume of nectar that each of these plants are providing for them, right? So in the morning, there's not much of a difference, so they can have it either way. They're like, drought or adequate, either way is fine with me. Whereas in the afternoon, they see that the adequates are providing more nectar, and so they're returning to those flowers more. And we believe that the higher drought tolerance of Phacelia that I told you about 10 minutes ago might be leading to these differences in response of Phacelia versus Nemophila to drought, and then thus the different behavioral responses of osmia to the drought in the morning versus. So to summarize our results for question 2, we showed that in Nemophila in the morning, bees tended to prefer adequate watered flowers, and they, again, in the afternoon, preferred adequate watered flowers. However, in the afternoon, bees didn't have much of a preference for adequate or droughted flowers. Oh, sorry, in the morning, yeah. And then in the afternoon, they showed a strong preference for adequate watered flowers. We think that these patterns have some important implications at both the species level and at the community level. At the species level, we think that if plants are growing on a kind of a heterogeneous landscape with different amounts of soil moisture, drought could decrease the seed set of plants with limited water by altering rates of pollination. We already know that drought can decrease rates of seed set just naturally by affecting the plant's physiology, but it looks like there might also be this mutualism mediated component of And at the community level, we think that drought could differentially impact the pollination of plant species depending on how drought tolerant those species are. So if we see two plant species co-flowering and one is more drought tolerant, that more drought tolerant species may be preferred by pollinators, whereas in a non-drought setting, that may not be. So to close out, I've got 3 take-home messages for you all. First, we know that drought can negatively impact flower number and size and also nectar volume. And we found that osmia tends to prefer the flowers of adequate water plants, but this preference increased in the afternoon for our more drought tolerant species. And overall, our implications were that drought conditions could decrease seed set by impacting floral traits and impacting pollinator preference, and that co-flowering drought tolerant and drought sensitive plants could result in changes to community composition. So with that, I'd like to thank you all for being here. It's been great to talk to you today. I'd like to thank my committee members, and in particular, for Rafferty, Santiago, and Cindy Glassman's lab. I'd also really like to thank the Shipley's Inner Lake Matthews Estelle Mountain Reserve Endowment, which provided the funding for this project, as well as the Label Award and the Edge Institute, which also helped provide funding. I had 8 amazing undergraduate students who helped me in the greenhouse and in the lab thereafter. In particular, Kim and Mahanor have been so amazing, and they've each received funding to pursue their own research in the same system. So that's super exciting. And finally, thanks to all the bees who helped me out, and some of them didn't make it. So yeah, really appreciate them. Thank you. And finally, yeah, I'd love to take any questions. If we don't make it through everyone's questions, please feel free to contact me at my email, which is listed there, or feel free to visit my website to check out more of my research. Yeah, thanks everybody. Thank you.
That was great, Annika. This is Katie. I don't have any questions in the Q&A right now. If anybody wants to post a question, go ahead and do that, or we can also come back at the end and see if there are questions for Annika.
Question in the room. So I apparently wasn't paying attention well enough for the water potentials. Just back in, yeah, there's a couple things that you said that were just kind of I think that was trying. Yeah,
Absolutely. Yes, leaf water potential. Let's try.
Can you close the chat?
Yeah, I'm gonna pop it over here to see. So leaf water potential is, in scientific terms, it's a metric of how much tension there is in the water column in the plant. The more tension there is, that means that there's more, that the plant is more drought stressed. Okay. And so essentially, as this number becomes more negative, that means that the plant is more drought stressed. Okay. So the closer these numbers are to 0, that means the plant's less stressed. And so what this figure was showing us is that the adequate water plants were across the board less drought stressed than the droughted plants. Yeah. Does that cover your?
That does. I
Okay.
just don't want the term to go on.
Yeah, no worries. It's a great. I hadn't thought about measuring this, but then Santiago suggested that I actually measure this to make sure that my treatment was effective. Yeah, so it's a nice novel component. I learned a lot. It is, yeah.
So the flowers under drought stress were smaller and had less nectar, right? So to this bee, that might not be as good, but in natural environments, there's a wide variety of pollinators, including smaller pollinators who might prefer smaller flowers and don't need the same amount of nectar. Do you think that this could spur like a shift in the pollinator community that visits these flowers?
Yeah, potentially. So I think, so this was Elijah's question. I think that they're in the desert in particular, so this might be most pertinent for like Phacelia campaniularia, which is more of a desert species. Little tiny perdita bees, for example, are a genus that are really, really small, and they might have display a slight preference for smaller flowers, which Phacelia are producing. But I will say that when Phacelia is really, really drought stressed, and when Nemophila is really, really drought stressed, it just produces like zero nectar, which I think is not preferable to any pollinator necessarily. So that's something I think that would need to be investigated further. But that's a great question.
There is a question in the Q&A, and then I'll.
Yeah.
Okay, great, thank you. Yeah, I'll address the question in the Q&A. So for folks in the audience, the question is if I could elaborate on the methods to measure nectar production. Yeah, I'd love to. It's one of my favorite techniques. It's really cool. We just use these tiny glass straws that are called microcapillary tubes. And they're each of like they can suck up a known amount of fluid. So like, for instance, you might have a 1 microliter microcapillary tube. And you take this tube and you just place it in the flower, like you're a pollinator yourself, sucking up that nectar, and the tube sucks up the nectar. And then you measure with a tiny ruler or a calipers. Yeah. You measure the length of nectar that's in that tube itself, and that'll tell you the volume of nectar. And then you can extrude that nectar onto what's called a refractometer. And it's this little analog machine that uses refractometry to tell you how much sugar is dissolved. Was that an appropriate answer to your question? Okay, Arlene.
My question may be a good follow-up. So there were different nectar concentrations as well. And like hummingbirds have a real preference for different concentrations of nectar. I mean, higher sugar content in the nectar. What's known about the different bees? Some bees actually be more attracted to the higher concentration of sugar in some of the drought stressed plants.
Yeah, that's a terrific question. So if folks in the audience couldn't hear, Arlie was asking how different types of bees might respond to different nectar concentrations. So yeah, I think it largely just depends on the physiology of that bee. If they're, I can see that larger bodied bees might prefer to have like a larger volume of nectar, whereas the tinier bees might be seeking out more concentrated nectar, but I don't have any evidence for that. I don't know enough about which bees prefer which types of nectar, but I think that would be a really good like future study to kind of compare which types of bees prefer which types of nectar and how that could shift what pollinator communities are visiting what flowers under drought conditions.
I have another
Okay, yeah,
yeah, it's more sort of bee biology oriented, but related to the pollination. So when it's hot, as it warms up during the day, and the air is getting drier, potentially might there be an attempt for some species to seek out more liquid in lower concentrations in order to keep their hydration up, I know. So I don't know what's known about that sort of detail in the biology of bees. So could that explain some differences with some bees foraging in the morning compared to the afternoon? If there are different preferences or different, so again, different concentrations, amounts of liquid available to them to bring back to their cells.
Right, that's
or just to keep themselves hydrated.
Right, and it might also depend on like the temperature, right?
Yeah.
So you're talking about like temperature from the morning to the afternoon,
Right.
but it could also be from June to August or something
Yeah.
like that. So that's a really great question, and I think that again, I think that we need to do more work on that. I'm sure that some folks have. I don't know if there are any like phylogenetic patterns linking like certain, if some genera need more water in the afternoon or something like that, but that would be really cool. My lab group, Nicole Rafferty, and her colleagues like Natasha Mancourt and Alessandra Fazzani, they worked on pairing drought and heat treatments. So I think that would be a good way to kind of look at those questions. And so, yeah, looking at how temperature and drought act together to impact not only plant physiology, but bee physiology, which will help us address those things.
Open question.
Okay, well, thanks so much, everyone. Yeah.
Oh, in the chat.
Stay there.
I was just going to say there aren't any other questions, so thank you.
Great. So much.
And so Katie, are you able to hear the audio, the questions from the audience?
Yes, just fine. Yeah, we should all be able to hear.
Great. Thank you. That was really cool.
All right, thanks, everyone. I'll hand it off to Annika.
It was in the same place. It's there, and then you just go. Go hide loading it. And
Steve.
Hi, everybody. Thanks for joining us here today. My name is Elijah Hall. I'm a fifth-year PhD student in the same lab that Annika is in. I'm in Nicole Rafferty's lab at UC Riverside. And today I'm going to talk a little bit about my project on plant-pollinator interactions from cactus to clouds. So first, I'm going to get into just a little introduction on the motivations of this study. As you may know, and from what Annika said a little bit, she did a great job introducing what I also talk about. So you will just flow right into it. So climate change is altering when and where species exist. A bee, for example, may have lived in the San Jacinto Mountains at 4,000 feet in elevation, and that's its historical habitat. But due to increasing temperature and altered precipitation patterns, in order to maintain the historical habitat, it will likely need to move upwards in elevation and potentially upwards in latitude. And in addition, it needs to change its phenology, which is the seasonal timing of biological events. So when bees become adults, they do that typically in response to a certain temperature trigger or a precipitation trigger. And because of climate change, those triggers are changing in terms of the timing of the year. And because of these different impacts of shifting temperature and precipitation patterns, species-specific shifts will mean that cohabitation in environments and interspecific interactions, so interactions between bees and pollinators, are likely to change, and they may not change synchronously. So flowering plants and pollinators need to respond to climate change in the same way in order to maintain their interactions. Sorry, I forgot to set a. And that can cause what are called phenological mismatches, when a flower blooms at a time of year that there is not a pollinator to visit it and pollinate it, which results in no successful pollination and reproduction from the plant perspective, and then no food resources from the pollinator perspective. So this ultimately negatively impacts plant communities, which we know are the basis of terrestrial environments, and ultimately can threaten ecosystem resilience as plant reproduction is needed in order to maintain the ecosystems, whether they be agricultural or natural. So studying the ecological interactions of plants and pollinators along elevational gradients, so from low elevation to high elevation, can provide insights into how climate change impacts ecosystems and how it will continue to impact ecosystems in the future, because an elevational gradient basically acts like a manipulation study, where as you go higher in elevation, the temperature gets cooler, and precipitation patterns change. So it's a space for time substitution is what we call it. So with that kind of background, I'll tell you guys a little bit about my project. So in 2021 and 2022, my collaborators and I collected plant-pollinator interaction data along a 7,000-foot elevational gradient to understand how plants and pollinators are responding to the ongoing drought in California along this elevation gradient. So we collected data on the plant-pollinator interaction network structure. So we looked at the whole plant community, the whole pollinator community, and then we measured how synchronous their phenology was in a whole bunch of different aspects about the interactions and how they're shifting through time and space. So the goal of our project is generally to predict how resilient pollination will be to future aridification in dryland ecosystems. It's kind of a large project with many facets, but today I'm just going to kind of be sharing the cool parts, which are just a brief look at the interactions through time and space, and kind of a visual summary of the research. So I don't have any cool results or anything to share. I'm just gonna show you guys the cool plants that I've studied. So a little bit about the study site that we use, the elevational gradient. So our elevational gradient begins in Palm Desert, and then travels up the Santa Rosa Mountains in the San Jacinto National Monument, and ends up on Toro Peak. So a lot of this work is based out of the Boyd Deep Canyon Desert Research Center, which is a UC Riverside Natural Reserve. And that's the base of our study, and then we go up in elevation. So I'll kind of introduce the sites that we have. So our lowest elevation site was at 948 feet in elevation at the Boyd Deep Canyon Research Center. And this here canyon, that's Deep Canyon, so that's where it gets its name from. And then this is the Santa Rosa Mountains here, which you can see from Palm Desert. And then if you're familiar with the Anza Borrego, they're the mountain range to the north. So our sites begin at what we call the campground. We have like colloquial names for all of our sites. So our lowest elevation is at the campground at Boyd Deep Canyon. And this is in the wash of Deep Canyon. So it's low desert and has species that you would expect at the low desert, such as creosote bush, tuberosa, palo verde, stuff like that. Our next site is called Agave Hill. So this is going up the mountain. So 3 of our sites travel right along Highway 74, if you're familiar with that. And this is a high desert site. So it's dominated by agave, given its name, and then also a large variety of cacti and other bushes, such as acacia. Our third site is called Pinyon Flat. It's in the community of Pinyon Flat. This is another site that is hosted by the Boyd Deep Canyon Desert Research Center. And this is in a pinyon juniper transition zone. So there are some desert plants, and then there are some, kind of chaparral transition plants as well. And then we move up into the mountains. So our next site is on Santa Rosa Mountain in the Chaparral, coastal Chaparral. So this is dominated by chaparral plants like manzanita and different oak species. And then lastly, we have our site on Toro Peak, which is in Jeffrey Pine Forest, all the way at the highest elevation that we can get to. And these sites are a part of a long-standing research project that has a total of 21 transects. So the campground is transect 1, and you'll notice that it shifts from saying campground Agave Hill to numbers, and those are a part of this historical group of transects that have been examining plant community composition for the last, I think it's 50 years now. A transect is just a straight line. So I'll actually talk about that right now. So at each site, we have 3 100-meter transects. So just 3 100-meter lines in space. So here is our first transect right here, and then our second transect, and then our third transect. And these are at our second site, Agave Hill. And so we use these 3 transects to conduct standardized collection methods, where we conducted 4 plant pollinator surveys each day. So along each transect, we walked it over a 20-minute period, looking for pollinators landing on plants. And anytime a pollinator landed on plants, or a flower that is, we tried our very best to catch that pollinator. And that's where we get our data on which pollinator is interacting with which plant. So we did that 4 times a day. And then in addition to that, we used 1 meter quadrats, so just a 1 meter square made out of PVC. And we collected 30 random samples along the transects twice a day. So we just kind of chucked the quadrat in a random direction, and then counted however many flowers are in that quadrat. And we did that a total of 60 times each. So in total, we conducted 52 surveys in 2021 and 56 surveys in 2022. And it totaled 432 hours of sampling along the transects, 20 minutes at a time. And yeah, it took a huge amount of work. And from those efforts, we collected over 3,800 plant pollinator interactions. So a little bit about the plants and pollinators that we collected. We observed 111 plant species that were flowering. And of those 111 species, 72 of them were visited by pollinators. So 39 of them were flowering, and either they were never visited by pollinators, or we didn't see them being visited by pollinators. And in addition to that, we collected 403 species of pollinators. So over the last year, my collaborators and I'll go back a slide. This is Natasha DeManicourt, who was a postdoc in our lab, and her, along with Alessandro Fizzoni, were my collaborators on this, and we collected these data together. So over the last year, we worked with the UCR Entomological Museum and identified the 403 species that we collected. So really a tremendous amount of diversity from both the plant and pollinator perspective. I study phenology mostly, so I thought I would show you guys what the phenology looks like at these different sites. So just to orient yourself to these graphs here, on the x-axis is time. So we started sampling at the lowest elevation in February, and then we stopped sampling at the highest elevation in mid-August. So you can kind of see each site by where or how they start. So the red line is transect, or is 1, which is the lowest transect. It flowered the earliest, and then the kind of green-yellow line. Trying to find the cursor is the second elevation site. The bright green line is the pinyon juniper transition site. The blue line is the chaparral site, and then the pink line here is the Jeffrey pine forest site. And on the y-axis is just the number of flowers. So you can see that the number of flowers was very low at the lowest elevation in both years, and pretty consistent between elevations in 2021. And then, as you can see, we kind of had an explosion of flowers at our third elevation site in the pinyon juniper transition zone in 2022, which I'll show you guys a little bit in a second here. So now I'm gonna kind of break down each site and show you the major flowers that were there, the major plants that were flowering, and then talk a little bit about which pollinators we're visiting. And yeah, so I'll get right into that. So this figure that we can see here is a network of all of the plant species that all 100 or all 74 plant species that were visited by pollinators on the left side here. And then on the right side is the other half of the network with pollinators. So this group is flies, this group is wild bees, we had some butterflies, we had our fewest number were observed hummingbirds, and then this large chunk down here were honeybees. So this just shows you, this isn't like legible or anything. It's not, it's hard to kind of understand these without looking at them very closely, but just to show you that the networks are quite diverse, and this is kind of the data that we use as researchers to untangle how the impacts of climate change are really affecting these plants and pollinators. Okay, so I'll start with the low desert site and its interactions. So this was the least diverse site among the 5 different locations that we collected data. And as you can see, the primary plant being pollinated there was Justicia californica. So that's tuberosa. And then the other major flowering plants that were visited by pollinators were desert lavender, creosote bush, Lauria tridentata, and palo verde. So just kind of a summary of the interactions there. The low desert site was dominated by large flowering bushes, and that's likely due to their ability to access water at deeper, at more depth underground. There were very few annual plants and few native pollinators. So all of these plants were most strongly visited by honeybees. Justicia was also visited by hummingbirds, as you can probably tell if you know about floral morphology, but red flowers are typically hummingbird pollinated. And they were also visited by a number of sweat bees in the family Halictidae. So there were some native bees that visited as well, especially on palo verde, the bright yellow flower here, which you're probably familiar with, was visited by a group of bees called centrists, which are large and extremely fast. So if you see a Palo Verde in bloom, you can probably, if you watch it long enough, find pollinators that or bees that really zip all around it and are very hard to catch but very interesting little bees. Okay, so I'll move on to the high desert site. So this was actually our most florally diverse site along the 5 transects. And the major pollinators or the major plants that were visited by pollinators were acacia. There was also a lot of pollination for agave. There's a lot of ocotillo there, so they were pollinated quite a bit. Bebia, and there was a large diversity of cacti as well. So these are some of my favorite plants at this site. And this one again was visited by a lot of honeybees, especially the acacia here down in the bottom center is a major resource of pollen for honeybees. And then the agave and ocotillo are not visited by honeybees. They're pollinated by a pretty diverse community of very small sweat bees in the family Halictidae. And then cacti are visited by various native bees as well, that they have a strong coevolution with. Okay, I'll kind of speed up here a little bit. So next is the pinyon juniper transition. So this is kind of my favorite site, and it's all because of our first species here, Remocaria lipida, which is it's formerly in the Cryptantha genus. And this site was really special because in 2022 there were blankets of little tiny annual plants. So we have the euphorbia in the pictures here. You can see the little sand mats euphorbia. The little tiny daisies are Areophyllum wallacei, and then our the little Cryptantha, and our other major blanket flower was Phacelia fremontii. And these were pollinated by a huge diversity of pollinators, including flies, lots of flies, honeybees, native bees. And at this site, we actually found a species of bee that has never been identified before. So there's actually a novel species here. Next is the chaparral. So this again, dominant species was the Remocaria lipida, as well as Manzanita, Trichostemma parishii, which is one of my favorite flowers, and then Ariastrum species. So here we had less dense blankets of flowers, but still quite a bit of floral diversity. The bottom right picture here is the Trichostemma. It has a really long, really beautiful and unique floral morphology. It smells great. So if you're ever in the chaparral, I recommend finding this. This is the shortest flowering season, and it's unique because it's dominated by kind of mostly non-flowering chaparral bushes. So a lot of these species just live in the gaps between. And then lastly is the Jeffrey pine forest. So here we had, this was actually our highest diversity of pollinators. Pollinator diversity increased as we went up in elevation. And it was just dominated by lots of beautiful plants like salvia and riboserium, and a bunch of Areophyllum species as well. So I'm running out of time here, so I'll just talk about I'll talk about the impacts of drought on these ecosystems. So the drought along this elevational gradient hasn't impacted the sites in the same way. So because they haven't received the same amount of precipitation. So precipitation increased as we went higher in elevation. These graphs show volumetric water content, so soil moisture. So as you can see, the lowest site had very, very low soil moisture, and it never had an early season peak like you can see from the other sites. And while that wasn't the case either at the highest elevations, they receive monsoonal precipitation that the upper two elevations do, unlike the bottom three sites. And what this has is a deleterious effect on pollinator flowering abundance and diversity. So here are figures showing the distribution of flowering species at Site 1, the campground, in 2021 on the left and 2022 on the right. So each different shape here is a flowering species, and with time on the x-axis and abundance on the y-axis. So you can see that even though the soil moisture was similar between the 2 years, the successive years of drought have caused a decrease in floral abundance at this site. And this effect is reduced as you go higher in elevation. And then here is the pollinator, the same thing, but with pollinators at the bottom site. So 2021 on the left, 2022 on the right. And as you can see, another successive year of drought had a huge impact and caused a major decrease in the number of pollinators in 2022. So we caught, I think it was like no more than 10 pollinators over like eight surveys at the bottom site. And that's because there was the good precipitation year in 2019, but 2020 was very dry. 2021 was also very dry. So the more successive years of drought, the greater the impact on plant and pollinator abundance. So generally, this does not bode well for the future of plant-pollinator interactions in dryland ecosystems. Long-term drought will negatively impact floral abundance and pollination. And right now that's happening most at the lower elevations, but based on our elevational gradient, that's likely to increase at higher elevations as well. And ultimately, there will be winners and losers of climate change. So some species will be able to exist still, but the diversity of both plants and pollinators is likely to decrease. And with that, I will take any questions and just kind of highlight some of my cohabitants out at the desert, which ended up being kind of a nemesis of mine, the bighorn sheep, because they love to eat flowers. And yeah, I just want to thank my lab mates, especially Natasha and Alessandro, who were out there sweating with me in the field, collecting all this data. And yeah, I'll take it. Yes.
So we don't have any questions in the Q&A, but maybe in the room.
Okay, I have a question. I wonder every week to capture that information. How much do we know about the influence of that repeated extensive sampling on the next season?
Yeah.
Future years.
Yeah, that's a good question, and definitely something.
Elijah?
Yes.
Oh,
Hi,
yeah.
sorry. Could you repeat the question? The sound wasn't very good on the remote side.
So
Thanks.
the question was, should we be concerned about the impacts of repeated sampling in these systems and how it might impact pollinator abundance? So definitely a question that warrants concern, like I said, and there's one study that's been conducted at the Rocky Mountain Biological Laboratory in Colorado that showed that repeated lethal sampling of pollinators did not have an impact on pollinator communities, so abundance or diversity. And that's probably not exactly the same here, but I will add that we only collected pollinators once a week, and so the total sampling effort, if you think about like is like very, very small. Like when we went out each day, we caught on average like 20 pollinators. So there are, just that we saw are like hundreds of pollinators that are outside of our transect bounds and stuff like that. So yeah, I don't think that our sampling probably had a negative effect, but I had like a bias because I don't want to have a negative effect, but we designed it so that we're not going out and capturing thousands of pollinators. No pan trapping, nothing like that. Yeah, that's how you can probably catch a lot of. Yeah.
So I have a question. I'll stand up so I can be louder. And how did you keep from spooking the pollinators along the transect? I mean, I can imagine if I were walking along the transect, I would see everything.
Yeah. Did you guys hear that in the chat? So the question was, how do I keep from scaring the pollinators away? So I've done this a lot, and so you just have techniques like you don't want to throw your shade over anything. Shade is the #1 thing. So yeah, you just have to be careful about that. And yeah, I guess I just have a technique that I've developed, like carrying my net around, being very sensitive to where they are. And I'll also note that like flies are pretty skittish, but bees are very much like in a one-track mindset to visit flowers. And yeah, so that's kind of the case. And also our catch rate is far from perfect. So like at Agave Hill, the second site, I had a very frustrating year in 2021 because I just, for the life of me, I had a really hard time catching pollinators from cacti because the bees that visit cacti are very, very fast. So, and you don't want to like destroy the cactus. So yeah, it's a challenge, definitely a challenge. Yeah.
No one they're going to hear me, so I don't have to repeat it.
Yeah.
But you were mentioning you collected a novel.
Yeah.
Is it novel for this area or globally? And if so, do you get?
Yeah. So the question was in reference to the novel bee species that we collected at the Pinyon Juniper transition site. And so, yeah, that had never been identified before anywhere. So it's a brand new species that needs to be named. And we work with the entomologists in the entomology museum. So basically we found that bee, and then Doug Yanega in the museum was like, we have never seen this bee before, and he's an expert on it. So we only know it's a new species because he told us. And then we found it in 2021 and then in 2022, Doug and his crew went out to collect more of them. So it's up to him. So if he is, I don't think he'll name it after me, but I was the one that caught it. So I don't know. There's a small chance that I.
One question? I'd just like to thank really interesting. Hopefully we'll get more people. I thank the audience, the Zoom audience, for being there, and Katie for hosting, and Orchid for helping to get our programs running. Thank you so much. So I guess we can zoom out. Wait, so Katie, you ready to end the program?
Yes, thanks everybody. That was a great couple talks, Annika and Elijah. Thank you. Wonderful.
Yeah, thank you for having us.
I guess we can.
Yeah, if you want.
And we did get some things.
Oh, good.